population dynamics. Okay, 1 second. Population dynamics. It's the title of this last unit. We're going to sort of go through, and basically it's an introduction to how this kind of thing is done. It's an ecological biology branch. It's not the biochemical, typical anatomy, physiology that we do. It's kind of a break from the usual. But, you know, it is an important branch of biology, managing populations of wildlife, studying populations, and all of those kinds of things that go along with it. So first, what we're going to do is just look at some terms and words, vocabulary, right, to learn the language of population dynamics. All right. So obviously, a population, you'll remember from, is referring to the number of individuals belonging to a single species. The word population is always talking about a single species. So the population of foxes, the population of humans, the population of maple trees, the population of bracket fungi, the population of some individual species in a particular area. And how those populations are affected and how they change and move up and down is essentially what the population dynamics is all about. And you probably remember that from grade 9. Okay. One of the things that we like to discuss before we get into numbers is how populations are distributed over certain areas. So population distribution is sort of described in some general ways. Now remember, these are generalized ideas, and there's always exceptions, and Mother Nature never follows our rules nicely. But these are just sort of some general frameworks in which we tend to work. So basically, if we look at the geographic range of a particular species, what this is referring to is the total area over which it is spread. Okay? The total area occupied by this population. Occupied by a population. And you might think, okay, well, isn't that just pretty straightforward? Well, it is, but you have to remember that if you're studying these things in detail, the total area over which a population is spread might not be the best way to look at it. Because within that area, there might be large portions of that area where the population doesn't live. For instance, a giant rock that's in the area, right? And then you have to look at, if it's a large area, how is the population sort of spread around in that area? So even though the geographic range, which is usually a wide-ranging, if you take, for instance, some North American songbirds, well, here's North America. Right? Basically, you'd have, a certain songbird might range all through this region. This might be the geographic range of this particular songbird. And that would be sort of a large area. But within that range, the songbird might not be completely evenly spread out. So we have to think about how we have sort of how it's spread. So what we can talk about is the crude density. Oh, well, density, population density then, isn't the number, it's the number of individuals in the species divided by or compared to the area, right? Compared to the area. Now, if you use the geographic area, you might say that, this particular blue spot on the map contains, 2.4 million yellow warblers, possibly. But how they're distributed within there is kind of important too. So we often talk about what's called the ecological density and the crude density. So the crude density is when you compare to the area, I guess I could say the total area, the geographic range. Let me just rewrite that. The geographic range, total geographic range. Area of the total geographic range. But to be a little bit more specific, we have what's called the ecological density. And so in order to do that, you have to be a little more specific about those areas. It's a comparison to only the areas occupied by the species or by the population within the geographic range. Okay, so we might find, if we go back to the map, we might find that the yellow warblers in this map are actually not just spread evenly out. Maybe they're focused around areas like this in the Great Lake region, where there's certain yellow warblers kind of like, they like fields and marshes, you know, so they don't live along the Appalachian Mountains as much. There's a bunch over here, maybe there's a pocket of them here. And so within here, we would find that there are pockets, right, wherever they might be. And so we would have to consider those pockets only in order to figure out the density of the yellow warbler from a more precise ecological sense. So that's the difference, right, between those two. So it's a pretty straightforward thing. They're both the number of individuals, but they vary as to what kind of area we're using to compare them to. So there. The other thing that we should look at is how they're spread out within that area. So distributions, and again, I'll just draw some, let's say, some regions. So let's say we have, we do a sample, and we find that here's a big chunk of land or water or whatever it is that we're studying, and we see that we've studied, let's say this is the number of dandelions in maybe Ontario or Northern Ontario, or maybe your backyard. It could be a small area too. What we might find with dandelions is that they're basically just kind of all over the place, right? And we get kind of a random distribution. Just kind of randomly everywhere. But if we looked at a different kind of population, maybe we looked at dolphins in a particular part of the ocean, we might find that the dolphins, because we know their behavior is to swim around in little pods, right? We might find that the dolphins are sort of distributed in what we would call little clumps. So we can call this more of a clumped distribution. And in some cases, although in Mother Nature, these are the two, you know, these are sort of the two that are most often. I'm just going to erase these words here so I can draw. We can also find a uniform distribution, which is a little bit less common, I would say, where the population isn't random and it isn't clumped, but it's pretty much like spread out like this. This could result from a population of a particular species that competes with itself, for instance. And so there's always a certain distance away from other organisms or something like that. It could also be an artificial situation. If you go up to Hiawatha Park, you'll find that often as you're skiing there in the winter, you'll go through these trails and all the trees are in these lines. And that's because back in the day, they reforested that area and they would go down the line with their tree planter and they would plant everything specific number of feet apart. So it looks very almost unnatural, right? Because you got a tree, every tree exactly 10 meters apart, and they're all growing in nice, neat rows. That would be more of a uniform distribution. So it's a little bit different than random because there's kind of like there's a specific sort of pattern, but it's not clumped up. So these are the three sort of general ideas, right? These are all just kind of general patterns. But of course, like anything, there can be anything in between, right? So something might look a little bit uniform, but still be kind of clumped. What if you're clumped, but the clumps are in uniform distributions? I mean, there's all kinds of, possible derivations of that. But basically, we look at how they're dispersed in those three sort of ways. Okay, so that's basically how we describe the population's dispersion and what population density actually means, right? It's pretty straightforward. Another word that'll come up a lot is the word habitat, and I'm sure that you've learned that in grade 9, and we have an idea of what that is. So a habitat is sort of like the specific places within the population distribution, the geographic range, that are being occupied, right? They're a little more specific. Specific parts of the geographic range being occupied by a particular population. So, for instance, if I go back to my example about the yellow warbler, we said that the geographic range was that blue section all over North America. I'll go up there in a second when you're finished copying that down. But the individual habitats within that range is where the yellow warbler actually lives. For instance, a marshy area around the tip of a Great Lake or something like that. So the habitat is as a sort of more defined part of the range. And it's defined by its specific, what we call biotic and abiotic factors, right? So let's write that down too. This is defined by the biotic and abiotic factors that are present there. And you'll remember, I hope, from grade 9 that biotic means the living factors, and abiotic means the things that are not living. Abiotic and biotic factors. So, for instance, the biotic factors could be, if you're a yellow warbler, things like what kind of forest is growing there that you're flying around in, what kind of plant life, is present, what other parts of the food chain are going on in that particular area. Abiotic factors would be things like, what's the relative humidity of that? What's the characteristics of the soil? Is it rocky or sandy? Or, those are not living factors. They're factors that are more abiotic. And so if we think about a swamp, that's a good habitat because we know in our minds that a swamp has got lots of standing water. There's mushy, there's mud. We know what the swamp characteristics are. We can name off the abiotic factors. Lots of water, it's humid, very, very rich soils because there's usually a lot of decay and mud buildup. The humus layer, humus is a nice dip. Humus is the stuff on the ground that's rotting and decaying, right? Anyway, those would be the factors of a swamp. If we said desert, that's a different habitat, obviously. Now, you also have the word biome, which you learned in grade 9, and they kind of cross over. So if I say desert, well, that could be a biome, but it could also be a habitat. And again, all of these words are basically, the biologists who love to categorize things make labels for everything, but there's a lot of crossover, and you have to be aware of that. Okay? So habitats, I realize I spelled this without crossing the T. It's not a habital, it's a habitat. There we go. All right, so if I go back to that picture now that you've caught up, the little red areas might be the actual habitats where the yellow warbler's living. Okay. All right, so now that we understand a little bit about these populations, what we have to figure out is, how do we measure them? Now, part of the problem is you can't just go out and count. There might be some situations where you can do that. I guess if you wanted to know the population of dandelions in your backyard, you could spend 20 minutes and go out and count them all. But usually we're dealing with areas much bigger than that. So the Ministry of Natural Resources wants to know how many moose there are in large sections of northern Ontario, because of course they have to hand out moose tags for hunters, and they want to make sure they don't have too many moose being shot, which would affect the population too detrimentally. So they have to study this, and they can't go out and count all the moose. So typically, the way we do this is by doing something called sampling. Sampling, well, is very similar to what's done with the recent election in the US and the polls that come out before the election. You know, they'll call up a random number of people, ask them questions, and then they'll use that number of people they've contacted to represent the bigger population, because you can't ask everybody. It's the same idea. So polling is very similar to what we do in natural habitats with sampling. And we're basically going to look at a couple of different ways of sampling. Okay? So the first way we're going to look at is something called Quadrat sampling, which is a pretty easy way to do things. This is usually a good way to do when you have a static organism like a plant. This is a sort of a better, more effective way to do this sort of population measurement. So what a quadrat is, right? It's a square or a rectangle. It's a four-sided area. Usually we use squares and rectangles because then we can calculate their relationship to the greater area more easily. So a quadrat is simply a laid out area. Now the size of the quadrat depends. It could be a meter by a meter if you were counting something very small, like, I don't know, the number of insects in a particular area. But it might have to be, a quadrat might have to be a mile by a mile if you were counting something more, a mammal, that, the beavers or something that are spread out more. So the size of the quadrat varies depending on what it is you're studying. If you are studying bacteria, your quadrat could be an inch by an inch, right? Because bacteria are so small, that's like a gigantic area for them. So the size of the quadrat will vary depending on the organism that you're studying. But essentially what you do is you take this quadrat, all right? And so what I'm going to do is I'm going to draw here a larger area. Here's a big, large area. This might be northern Ontario, okay? And so this quadrat was taken from an area, maybe, I'm just going to draw little lines here. Maybe I'll use the dotted line. Oh, not the big dotted line. That looks terrible. Let me get rid of that. I'll use a smaller dotted line, if it will let me. Okay. So if we sort of take this quadrat might be taken from this spot right here. And then another quadrat would be taken over here. And so they would go through this large area of northern Ontario, and they would just delineate certain quadrats kind of in a pattern or somewhat randomly. And you'd have to take into account, of course, all the factors that we talked about before. If you're studying maple trees and you go and put this quadrant right on the top of a big barren rock, well, that's not really going to help you. It's going to skew your results because you're going to get 0 maple trees on the rock. So you have to be careful when you're selecting your quadrats that you're selecting a range of habitats, right? And so you put all these quadrats out there. And then what you can do, of course, is you can calculate the area of your quadrats. And then you can calculate, or you can compare that to the area of the entire blue section. So let's say in your quadrat, you counted, you were doing maple trees or something, and in your quadrat you had these maple trees like that. So you would count 1, 2, 3, 4, 5, 6, 7, 8. So you would say, well, this quadrat had 8 maple trees in it. And then over here you counted, and this one had 2 maple trees in it. And this one had 16 maple trees in it. And then you kept counting, and this one had 9. And maybe this one has 7. And this one has 11. This one had 21. This one had 2 again. And maybe this one had 5, or something like that. So you do this little sampling method, right, with your quadrats. And then you could say, okay, let's compare. So let's say you did quadrats that were 100 meters by 100 meters, right? That means one quadrat is 100 square meters, right? Or we could write, typically we would write that as meters squared like this, so I'll do it that way. 100 square meters. But this blue area is very large, right? It's very large. So your blue area might have an area of 250,000 square meters, let's just say. So what you do is you add up, you make a ratio, and on the top of your ratio, you put all of the numbers that you found of the population. So you add your, I'm going to have to go down here like this. Was there one up on top there? No. So all those green numbers we had, I'll do them in green so they match up. We found 8, then we found 2, then we found 7, then we found 9, and we found 16, then we found 11, then we found 21, and 5, and 2. So we're adding up all of the numbers that we found within our thing, a total number of individuals. So I'll just add those numbers up quickly. I think it comes out to 81. And then we have to add up how much area that is. And if you think about it, well, we have how many quadrats? We counted 1, 2, 3, 4, 5, 6, 7, 8, 9 of them. And they're all the same size. So that would mean on the bottom, our quadrats in black here would be 9 times 100 square meters. So the total area is basically 900 square meters. And if we just do the division, right? 81 divided by 900 gives us 0.09 per square meter. 0.09 per square meter. So far, so good. Yes. All right, somebody just pointed out my math mistake, which is, I'm glad you pointed it out now. If it's 100 by 100, it wouldn't be 100 square meters, it would be 10,000. So to fix that, we're going to change the size of our quadrant so all of our math remains the same. These should be 10 by 10, right? So let's just make these 10 by 10. I'll rewrite that number there. In my mind, I wanted it to be 100, and I screwed up on that. 10 by 10 is 100 square meters, right? 10 times 10, length times width for area. So that's what it should be in the quadrants. So these are very small quadrants. And then, of course, this number makes sense, because if you do 10 meters by 10 meters, they're not going to find 20 maple, and in fact, these numbers don't make sense for maple trees. You're not going to find 21 maple trees in a small area like that. So maybe this isn't maple trees, maybe it's smaller plants, like maybe it's dandelions or something. It doesn't really matter what it is. For the example, it's just showing you how to do the process. Okay? Now, 0.09 per square meter, right, is one way of looking at it. You could also look at it as how much area does one individual sort of take up. So you could flip that upside down, and if you did 90 divided by 81, right, 900 divided by 81, you get 11.1, which means that an individual is taking up about 11 square meters of space. But typically, we do the density this way. Now, it's possible that your quadrats could be different sizes, but if they're different sizes, you basically have to just account for that by adding up the proper area of your quadrants. Okay, so that means then, what's the estimate of the population over the whole? Well, now that we have this little estimate, we can say that the ratio of 0.09 for per square meter can help us estimate if now we have 250,000 square meters. We can figure out what number should go there so that it's in the same ratio, right? It's in the same ratio. And the easiest way to figure that out is to use some math tricks. Let's put a little n or x here. Let's put an n because we typically use n's for the number of individuals, the population. So let's put a little n here, n. And let's figure out what the number in the population would be. Well, if we remember this is a 1, right? 1 meter square, and we cross multiply, we would get 1 times n equals 0.09 times 250,000. And then we can do that. And it comes out to 22500, I think. I'll just double check the numbers to make sure. And this would be then what we would expect to find in the total blue area. So it's not difficult, is it? It's just cut it up in pieces. Count within the pieces the numbers of individuals. Compare that to the area, the total area of the pieces, and then get a ratio that you can then extend to the overall area. So what becomes a challenge is when you have animals that move, they're not necessarily static, so you have the danger of counting an animal in one quadrant and then going to another quadrant and the same animal has ran over there and you're counting it again. So in this case, we have another kind of system called you catch animals, you mark them, then you release them back into the environment. So this is called the mark and recapture sampling. And it's a little bit confusing, but once you get your head around it, it's not so bad. So here's what you do. I'll tell you what you do first. Okay? You have a whole bunch of animals in a region. So what you do is you go and you trap some, you capture them alive, and you mark them. You put a tag on them, you spray paint something on their fur, whatever. You put some kind of mark so you know that those were the ones that you caught. Then you release them back into the wild, and you wait a little while so that they'll mix and mingle throughout the whole area. Then you go back and you capture a bunch more. This time, though, you're probably going to capture some with a mark and some without, right? But the proportion of those marked to the ones that aren't in your second capture can give you a snapshot of the bigger picture. So that's kind of the idea. All right, so we need some letters here. So N is the total population estimate that we're trying to sort of figure out from our sample. But we are also going to have to capture some animals and mark them. So M is the number of animals that we capture and mark. So these are the number captured, marked, and released. Okay? Little n is going to be our second go out and capture. So that's our first capture is the big M. We captured a bunch, we marked them, and we let them go. Then we go out again and we capture another bunch. So this is the second capture number. And of course, this time we'll have a mix because some of them will have a mark and some of them won't, right? And then little m would be the number marked in the second capture. Okay? So in a sense, those in your textbook calls those the recaptures. Those have been captured twice. They were captured once because you marked them, and now you captured them again. So they sometimes refer to those as the recaptures. Okay? And so what we can do is we can say that in our first estimate, we have the total number that we marked, and we're going to compare that to the total number of individuals we think are in the entire area. But that ratio should be similar to the number that are marked in our recapture compared to the number that we have in the second capture. That's the second capture is like the sample. So you've marked them all and let them go to set it up, and then you take your sample, which is the second capture. And within that, there'll be a nice distribution. There'll be some marked and some not, but that proportion, if the animals have spread out evenly, should represent the same proportions as the big area, right? The total mark that are still out there. Now, what we like to do is resolve this so that it's solving for the capital N, which is the estimate. And you can just do that with some simple math. So over here, if you take that and you cross multiply, you'll get Mn equals big N, little m, right? And then if you divide both sides by little m, you will get a nice number, a nice equation that gives you the estimated population size. And this is called the mark and recapture method. Okay? So for this one, because it's a little bit confusing, we'll do an example of how this would work. All right, so we're going to do an example of the mark recapture sampling. So we'll just use the example in your textbook. They're doing the saw-whet owl, which is a tiny little owl. It's very cute. If you Google a picture of the saw-whet owl, you will fall in love with it. Saw-whet owl. I believe it's the smallest owl. So biologists are studying this. So the capture and mark number, right? And remember, we'll be going back here where we're using this little formula. So the capture and mark number, that's the capital M, right? So capital M. They went out and they captured and they marked 30. 30 of these saw-whet owls. They all have a little mark on them of some kind. Then they released them. And they waited. You have to wait, obviously, although you can't just release them and go, because you have to give these marked individuals time to sort of disperse and become more randomized throughout the greater population for the next part to work. So there has to be a time lag, right? Okay, then they did a second capture. The second capture is the small n, right? The small n. And they found out in the second capture, they captured 80 individuals, and they discovered that within that second capture, we find there's a small number that are marked, small m. There was 12 individuals marked. So you can think that 12 marked out of the 80 is a proportion that should reflect the number marked originally out of the entire population, right? And so we can figure out, if we go back to our equation right here, capital M, small n, over small m, right? So n equals capital M, small n, over small m. Don't confuse them. Make sure you get them right. And so we plug in the numbers, and we would get our capital M, which is 30, And then our recapture number is 80. And we're dividing that by 12. And that gives us an estimate. So if we do 30 times 80 equals divided by 12, we get about 200, I think. So it equals 200. Right? And if you look back at the original numbers, I'll just do a box here. We're basically saying that if we marked 30 out of 200 in the total population, that should be a ratio that's very close to 12 marked out of 80. That's basically what we're doing with that formula, right? This is the marked smaller sort of sample over here, and this represents the larger population. Oop, I think I have that upside down, sorry. I do have that upside down, don't I? 12 out of 80 would be, that should be 30 out of 200. I put an extra 0 on there for some reason, didn't I? 30 marked out of 200. So if you do 30 divided by 200, that's about 0.15. And if you do 12 divided by 80, I think you get the same number, 0.15. So both of these ratios work out to 0.15. So that's another way of thinking about it. If the formula gives you trouble, we're basically saying that the ratio of total marks to the total population will be similar to the ratio of marked in the second capture to the total in the second capture. You have a question over there? All right, so I guess I should just summarize. We estimate there are 200 saw-whet owls in this area based on the smaller capture size. We only had to capture 80 to figure that out. And that's really all there is to it. Now, of course, there are limitations to both of these methods, to the Quadrat method, and we talked about some of those limitations. We won't list them all, but just thinking about the quadrats, first of all, you have to make sure your quadrats are properly spaced out and randomized within the area. You can't do all your quadrats in a similar spot or habitat because that gives you a false reading. So you have to be selective in how you pick your quadrats. The size of your quadrat, right? So the position of the quadrats, maybe we should write some of these down. These are limitations to understand, if you're a biologist, things that might screw up your numbers, right? Limitations, things that you have to be careful of. The position of quadrats and how you pick the spots. It needs to be relatively random. Random across the area, including all the different possible habitats that might be within the area to get the best, or the most reliable numbers. You have to think about the size of the quadrats. I wrote quadrants up there, but it's quadrats. Quadrats. Think about the size. That's kind of very important too, because like we said, if you're doing elephants, you can't have a quadrat that's 3 feet by 3 feet. You have to have a quadrat that's quite large. Okay? No, it's just a quadrat. Quadrat, not a quadrant. Those are two major limitations of the quadrat sampling. And then the number of quadrats, I guess we should also consider that. Like if you only do 2 quadrats out of a whole area, and the area is very large, well, that's not enough. The larger the area you're sampling, the more samples you'll get. And the more quadrats you use, the closer you'll get to a better estimate. It's like a poll for an election. If you only poll 30 people out of 200 million, you're not going to get a very good indicator, right? So you want to do, each poll is like a quadrat. You want to poll these 30 people and then poll another 30 people and another 30 people and another 30 people. So you're getting more people's opinions. Same thing here, number of quadrats. And of course, if we look at the other option, the capture-release method and marking method, there's all kinds of issues there too that you have to be aware of. You have to make sure that your marked individuals, are randomized. So you have to be careful. Because remember, you're capturing these animals, right? To mark them. So if you're capturing animals, it's possible that you're only getting the easiest ones to capture. So when you capture again, they're the easiest ones to capture. You might capture more of the marked ones than you expected, which will give you a false indicator. So you have to be careful about the actual animals that you're catching. And you got to make sure that you're catching a variety and a random sample. If you're only catching all of the saw-whet owls with one wing because they can't fly away from you, then when you do your second capture, you're going to get all these one-wingers again. And it's going to make it sound like, oh, look, I got like 60 out of 65 were marked. That must mean there's tons of owls. And that just skews your results. So you have to be careful about the actual animals you're calculating or you're catching. The other thing is the mark. The mark has to last. If you mark these animals with something that rubs off too quickly, like, you can't mark them with a dab of butter because that'll rub off and then they're gone. So you have to use something that you're sure is going to last. Tags. If you're tagging animals, well, it can't be a tag that's going to fall off. And you have to be careful how you tag the animal so that's in a way that the animal doesn't pull it off, right? So they'll often tag the ears they tag cattle that way. They tag the ear. Pretty hard to bite something off your own ear, right? And you don't necessarily rub it off too much. But animals aren't going to be agreeable to having tags and things stuck on them, so they will probably pull them off if they can. The time. You have to be careful of that timeline. The time between captures. You have to allow for enough time for them to spread, or else you're just going to go capture the ones you just released because they haven't gotten away from you yet. They haven't mingled in the population, right? The other thing about the marks have to last, but the marks can't be hazardous. Because if you're using a mark that's killing the animals, well, then that's going to skew things too, isn't it? So whatever you're using to mark these animals has to be something innocuous that isn't going to give them a survival disadvantage, and then they're all going to die. And then you're going out and you're like, oh, I'm not catching any of the marked animals. I wonder what happened. I guess there's not many out here. They're all dead. So there's all these limitations that you have to be aware of. But that's basically the concept of population dynamics, measuring populations, population densities using numbers and areas. And we'll stop there.